Bom dia, obrigado pelo convite para falar hoje. The current pandemic has really raised the attention to the link between biodiversity and pandemics. And it's also shown the susceptibility of wildlife to human disease. But we've seen cases like this for many years in many places. My own work has focused on these efforts across the tropics, really focused on land use change and the interface between wild primates and people and their domesticated animals, as well as vectors as intermediaries in some cases. Really trying to understand both sides of this equation of our pathogens impacting wild primates and the potential for health-related issues linked to zoonoses from primates to people. There's a great short film called We Are Nature that really highlights this intimate relationship that we have with natural systems, especially those biodiverse systems. And I just want to show you the trailer quickly to give you a taste of this.
In early 2020, we found ourselves in the midst of a global pandemic. Nobody knew where the virus had come from. But one thing was certain: somehow, somewhere, it began with us.
There's so much out there in the natural world. We're part of it, and we rely on it.
And because we're encroaching into wildlife habitat and traveling at an unprecedented rate around the planet and consuming products, meat, agriculture, livestock production, deforestation, road building, and that drives an increase in pandemic risk.
Wrecking the ecosystem, disturbing it, we expose ourselves to the viruses that the wild animals carry. Humans see themselves as detached from nature, as apart from nature, as above nature, and not as part of nature.
It was very convenient for many people to believe that animals were just things, just objects, to hunt them, shoot them, kill them, eat them. All of these things aren't so easy if you actually believe that you are dealing with a sentient being.
We have an obligation and responsibility to treat nature with respect and in recognition of our interconnectedness.
Not as an inanimate object, not as a commodity or something to extract from for human gain, but really something that reflects the health and well-being of humans.
We are part of nature. Nature has inherent value just as we have.
And we may think that nature is something separate and we can distance ourselves and live in our little bubble. It's not true.
How do we try to better understand these dynamics so that we can mitigate the impact of disease? Well, I'm going to talk about some examples from my own work to give you a sense of how we do this, because it's, like I said, the problems are complex. And so we have to really think outside of the box a lot of times to come up with solutions. So I primarily work in sub-Saharan Africa, Madagascar, and in all of these areas, we're seeing an expansion of the rural complex. There's more areas that are being cleared. We've talked about habitat fragmentation. We've talked about habitat loss to other activities. And so in this figure, you're seeing areas in dark green that are intact, tropical forest systems. And then in light green, you're seeing that rural complex. And that's a mix of subsistence agriculture. Sometimes it's only subsistence agriculture. And in other regions, it may be also including commercial activities, like monotypic stands of cash crops, plantations. So in Uganda, for example, you see that there's a lot of the rural complex, but very little remaining forest habitat. In that system, there's actually a mix of subsistence agriculture and a number of commercial activities like tea plantations and things along those lines. All right, so we work in those systems to actually look at how various types of land use change could lead to changes in disease dynamics in natural populations of wildlife, as well as how those changes might lead to our pathogens jumping into those species of concern. In our work, we also look at the flip side of that. We want to understand the global health related aspect and thinking about emerging diseases as part of the same dynamic, but we always typically try to integrate both sides of that equation. To get answers to these questions, we use an integrative research approach. So we're doing a lot of epidemiological and socioecological survey. So we're having stakeholder meetings, we're meeting with focal groups, we're doing interviews at the home. So that's one approach that we use. We link that to infectious disease diagnostics. So looking at various pathogens of concern or using model systems to understand transmission dynamics. We link that also to spatial analyses, so understanding not only what is happening in the system, but where it is happening, because oftentimes that can show us where there's a clustering of risks, both in terms of behavior and location for transmission between species. And observational data. We need to see what's happening. So what are people doing? What are the animals doing? And we think about the wildlife as well as some of the potential intermediaries like our domesticated animals. And it's important to remember that when we try to look at these things in wild species, we're often constrained in ways that we're not constrained to understand disease in people, right? So if someone is sick, they can go to their physician or another health provider, and they can explain what they're feeling, the symptoms that they're feeling. They can give details, they can respond to questions, and they will willingly provide samples that are needed. So a blood sample to be able to screen for something that might be impacting them. Or like as we're seeing with the coronavirus, taking a nasal swab, a deep nasal swab. We can't do that with wildlife. We can't get them to answer our questions about their health. They're not coming to us when they're feeling ill, and we're not going to get those in what we call invasive diagnostics, sticking with a needle to take a blood sample or doing a deep nasal swab. Those are things we cannot do for ethical reasons, but also logistically. For many reasons, we just can't do that. So we have to think about innovative, non-invasive ways to get answers to some of those questions. So that often means following these individuals of a species of concern at a safe distance, making sure we're taking precautions to protect them as we're collecting the data, observing them, seeing what they're doing, and seeing if there are any clinical symptoms that maybe we can just observe. Are they coughing? Do they have diarrhea? These are things that can help to get a sense of what could be going on. And then we're able to collect fecal samples from them. As they move out of an area after defecating, we can go and collect that sample. And fortunately, because we know individuals, we're able to link that sample to the individual. And because of the technologies available, we can also use various molecular techniques to confirm the identity of the individual. But it means it's a lot more work for us to figure out what's infecting them and what disease it's causing within their bodies, or even if it is causing a disease. Those are things that we're always trying to work out. Another component of that is that when an individual dies, we want to recover their body so that we can do a necropsy. So that's similar to an autopsy, but with wildlife. And getting a sense of, did they have pathology? Did they have conditions that were probably related to an infectious agent or to some other process? I mentioned the observational aspect of understanding what's going on with their health. Over the years, we've developed a number of different ways to do that, starting with paper-based forms and then moving to different things that can be done with a smartphone, for example. So now we have tablet and smartphone-based methodologies. And those are all now linked spatially. So we have spatially explicit information about health, and most of that, again, is observational. Another way that we get at understanding these pathogen dynamics in these systems is to use model systems for understanding transmission dynamics. So when you guys hear about E. coli, you're probably like most people, you think about romaine lettuce or some other outbreak of disease, foodborne illness that's been caused by a strain of E. coli. And that's typically how people think about E. coli around the world. If someone says E. coli, they're thinking, I don't want that. But the interesting thing is they already have it. We all have it. So there are strains of E. coli that are very pathogenic, that can cause foodborne illness, but that is the exception. Those are rare relative to the standard form of E. coli. This is a bacterium that we all have. Every mammal has E. coli as part of their microbiome, and they're actually beneficial to us. They help us to break down food. They provide some vitamin content in that process. They also help us to not become infected with some of the pathogenic forms. So when we knock out E. coli, along with everything else with a general spectrum antibiotic, that opens up the door for other pathogens to come in. So E. coli is important for our health. We all have it, and we have a misconception that it's something that's always bad because of these foodborne illnesses linked to certain strains that are pathogenic. Well, because it's so common, it actually serves as a really good tool for people like me who want to understand how easy it is for a pathogen to jump from one individual to another or one species to another, based on the kinds of interactions that they're having. And since all of the mammals have E. coli, if we can actually collect fecal samples from all the species of interest, we can then compare the E. coli isolates from those individuals to see how similar they are. And if they're very similar, it actually implicates a cross-species transmission. So we can actually look at those processes through a form of DNA fingerprinting. And that's actually what we've done in a system in Uganda, where we've looked at a number of species of wild primates and domesticated animals and people, and we've compared their E. coli isolates, just the run-of-the-mill commensal E. coli, the one that's not causing any problems for them. And we can look at how variable that E. coli is between these different species and from location to location, where there are different types of activities occurring that could increase or decrease the risk of cross-species transmission. And by doing that, we're able to see where we have strong similarities between the bacterial isolates, the E. coli isolates from these wild primates and people and domesticated animals. And so in the system in Uganda, we looked at this, we used something called an FST score, which is a measure of genetic distance. And basically the smaller the score, the more similar the bacterial isolates are. And what this shows in this figure is that there are three types of monkeys. Black and white colobus are BWC, red colobus are RC, and red-tailed guenons are RT. Those red-tailed guenons, as you can see in light gray, have bacterial isolates that are significantly more similar to the bacterial isolates from the people and from the livestock compared to what we're seeing with those two other monkey species. So that suggests that those monkeys are sharing this E. coli bacteria with people. Doesn't tell us the directionality, it just tells us it's very similar and they likely have been swapping it in some way. And that looks less likely with these two other monkey species. So then we need to start thinking, okay, why would that be? Can we get some other types of data that help us to understand what's happening? Well, we can bring in the spatial data to look at what's happening where. So by interviewing people and asking them about different activities, we can figure out, in here, in this figure in red, you're seeing where the house is, but then the other colored circles are the different types of activities they're involved in and where they go to do these things. Where do they go to get water? Where do they go to tend their cattle, etc. And then you're also seeing kind of the core area used by the monkeys in the black triangles. And then there are a couple of other things on the map here. You're seeing a lot of smaller dots in light gray and in light blue. Those actually represent another process in the system, and that is movement, and specifically movement of a certain species, in this case, dogs. So we decided we needed to track dogs to understand where they were going and what they were doing, because dogs can be really important intermediaries for disease transmission. We know that they hunt the monkeys. We know that they don't always stay around the house. And what you're seeing here are these two series of dot clusters. The one is a dog that does kind of stay more close to home, but you can see some of those dots go into the forest proper and overlap with the monkeys, whereas you have another that follows the forest edge, and you've got many other forms of movement of these dogs in the system. But understanding where they go and when they go there helps us to understand what role they might play in some of the transmission dynamics. And you might ask, well, how do you measure where these dogs are going? So we interview people and ask them where their dogs go. They don't typically have a really good sense of that. And it's not always the case that they know they don't have a sense of that. Many people may tell you they know where their dog is and what it does in your interview. But then what we do to actually verify that is put this small GPS unit on the dog. This I Got You unit is quite cost effective. They cost about $30. And the battery life is strong enough to last for a few days. So we basically duct tape these to dog collars and let the dogs go for a few days. And then we get them back, replace another collar to get more data for a few more days. And then we download all of that data. That's how we get to a scenario like this, where we have a better sense of where they go and what they do. We also have used dogs in other work that we're doing because of their amazing ability to smell. So you might be shocked to know, but we took this team of dogs that you're seeing here, trained sniffing dogs from an organization called Working Dogs for Conservation. They use the same ability that's used to sniff out drugs in airports or illegal produce that's being brought into airports, to sniff out other things of value. And in this case, what's valuable to us is gorilla poop. And these are gorillas that are so rare that, and they're hunted, so they're always hiding. The only way we can get their poop is to have the sniffing dogs help us. These are the Cross River gorillas, the most endangered of the apes. We did a 6-month expedition into this zone, rugged landscapes between Cameroon and Nigeria. And these dogs helped us tremendously to find all of the fecal material from the gorillas, which allowed us to do population genetics to figure out kind of how viable are the populations that remain of this critically endangered species, and to do an assessment of what pathogens they have, what's their baseline, and are any of those things that are likely coming to them from people or domesticated animals. So these again are ways we need to think out of the box to come to solutions. But that's a bit of a segue. Getting back to Uganda, getting back to thinking about the data that we had there, and this red-tailed guenon that was having bacteria that's very similar to people. So the E. coli is more or less like the people, but these other monkeys don't have that same phenomena. How do we get more of a sense of what's going on there? Well, another technique that we used is called a tally board approach. Here you can see one of my field assistants taking notes and meeting with a member of the community who had been collecting information about crop rating. So living near a forested area, animals may come out and eat things that you're growing for your own consumption. So people have their subsistence farms, and various wildlife species come and cause trouble. And by helping them to keep a record of what species were coming when, it helped us to get a better sense of where there was a potential for disease transmission as part of that process, but also to help people think about mitigation strategies, because they often would only remember, the elephants coming, because it would be one tragic event where a bunch of banana trees got smashed, or baboons coming, and again, just really going after their papayas, but they wouldn't notice the day-to-day impact of some of the other species. And what we found is that those turned out to be really important. So with the tally board approach, they're collecting information each day. So if something happens, they have a little picture of the animal they have pictures of many different animals and pictures of their crops. And each envelope has a picture of one of the crops they're growing. They typically do intercropping. And then they have all these different pictures of potential crop raiders. And if a crop raiding event occurs, they put that picture in the envelope corresponding to that crop. And they just write the day of the month on the back of the picture. And by doing that, we get a much better quality of data on crop raiding activity. And that gives us information like this. So here you're seeing the different species that turned out to be important in the system and the crops they were going after. And something that turned out to be really important, thinking about what we saw earlier with the E. coli and the red-tailed guenons, is this link to maize. Maize is corn. And as you can see, the red-tailed guenons are the most important crop raider of the corn. And you may say, well, why does that matter? Well, what we learned through those interviews is that people also were doing some mitigation of crop raiding already. They were taking cow poop and rubbing it on the ears of corn, sometimes with other things like cayenne pepper or sand, to try to deter crop raiding. And the challenge here is that we then were able to go out and witness what was happening. The monkeys, this actually worked. The monkeys would not end up taking that crop, that ear of corn, for example, but they still would investigate it. And so they would get the fecal contamination, and the people were also getting fecal contamination as they were applying the cattle feces to the ears of corn. And so this also, not only did this help us to understand how the red-tailed guenons were getting infected with the E. coli of people, it also helped us to explain why the E. coli was so similar between the people and the livestock. It was because the people were getting infected with the E. coli of the cattle in the same way, well, not in the same way, but in relation to this same mitigation strategy as the monkeys were getting infected. So that's an example of how, by collecting many different types of data, we get a much clearer sense of what's going on and how we might be able to improve outcomes compared to if we were just collecting one type of data. We've done similar work number of systems to look at how things like tourism can impact bacterial exchange, again, using E. coli as a model. And we've done this with gorillas and chimpanzees. And as you might guess, the more overlap gorillas have with people, the more overlap we see in the similarity of their E. coli. And again, in these systems, also in Uganda, we see a lot of overlap with the E. coli of people and the domesticated animals. So again, there's issues there that need to be examined related to hygiene and animal husbandry to try to reduce that exposure, because we know that, even though we're looking at E. coli that's non-pathogenic, we're looking at it specifically because it follows the same pathway for infection as a number of really nasty pathogens that we do have to worry about. Another thing that we've been able to do, kind of thinking out of the box, is look at antibiotic resistance in these species in relation to these factors of overlap. And this is important because you'll recall I said with E. coli, it tells us how similar the bacteria is, and it suggests swapping between species, but it doesn't tell us anything about directionality. We can infer that a bit through some of the other data streams, but one of the really good ways to look at this is by looking at where we find high rates of antibiotic resistant bacteria, because we're not giving antibiotics to the wildlife in most cases. And if we do, we have a record of it. So when we see resistance in populations where we don't expect it, because we haven't had past use of antibiotics, we then can more strongly infer the directionality of transmission. So the fact that these gorillas that have the highest level of interaction with people and have the bacteria that's the most similar to people have the bacteria that's the most similar. The fact that the antibiotics are being used in the people and the livestock, we can infer that that's then being transmitted already resistant to the gorilla populations. And then, as you can see, the gorillas that have less interaction with people and less similar bacterial isolates have lower rates of antibiotic resistance. But even the truly kind of wild gorillas that have more or less no potential exposure to human bacteria still have some level of resistance to antibiotics. And so if you think about that, there's an important evolutionary reason for that. And that is that resistance is basically a response to a selective pressure. And there is going to be overlap. They're going to be in natural systems, other selective pressures that are going to lead to some minimal level of resistance because that trait is linked to other traits. And so what's interesting is anywhere we go in the world, we always find this very low level of 4 to 5% resistance in species like Arctic foxes and the other most remote species. The other thing we were able to do is to see kind of where this resistance was coming from is link it to our survey data on what antibiotics people were using and the antibiotics that are available in their communities. So we use a tally board here, again, with a template of drugs. So you can ask people what drugs they take. They don't always know. Sometimes they know which colors it is, if it's a double color. We know sometimes people are taking antibiotics when they think they're taking non-antibiotics because of that, because they'll send a neighbor to an informal pharmacy and they'll say, I need double color, yellow red because they need a muscle relaxer. And they're actually taking an antibiotic because it has that same morphology. So we then also show them the morphologies as another way to try to figure out which drug they actually are using. And then we have them save the wrappers. And that's really informative, because not only do we figure out if it is truly the correct drug that they're taking, but we figure out where it was manufactured, if it's expired or not, and also if they're taking the full course of the antibiotic or taking just a few pills, which can then lead to greater rates of antibiotic resistance in the future. So what do we do when we want to look at an actual pathogen? So we talked about using a model system like E. coli. Well, in the systems where we've worked for longer and we have a good sense of kind of what's out there and we want to understand it better, we can do a targeted study to look at it. And so for the PhD research of Michelle Parsons, she did her PhD with me at Emory and is now at the CDC in the Global Health Division, we wanted to specifically look at cryptosporidium in that system because we knew that it was there. We had already seen that it seemed to be linked to some of these signs of illness. So we saw an association with diarrheal disease in them. And it's not common to find that kind of a linkage in wildlife populations. So we wanted to figure out, was this coming from people? Was this natural in the system? Was this coming from livestock? We saw all these interesting livestock linkages in Uganda. Was the same type of stuff happening in Tanzania? And then another important part of this is that the chimps at Gombe have a form of SIV, the simian immunodeficiency virus, that actually causes disease in them. And since cryptosporidium is a really important co-infection with HIV in people, we're concerned about it also as a potential cofactor for the impact that the SIV was having on this threatened population of chimpanzees. So we used molecular approaches to look at the cryptosporidium in the system, and we compared what we saw in the livestock with what we saw in the people and the wild primates, including the chimps and baboons. And what was interesting is we see 3 different forms of cryptosporidium in this system. So it's really complex. We've got people infected with cryptosporidium hominis in the park and outside the park. We've got the livestock, in this case, infected with cryptosporidium zowii, a totally different type. And then we have the chimpanzees and baboons infected with cryptosporidium hominis. So again, that may be linked to what we're seeing in the people. But then some of them are also infected with cryptosporidium suis, a form typically seen in pigs. But people in this region don't keep pigs. This is a Muslim region of Tanzania, and pigs are not kept as livestock. But there are bush pigs, as you see in this picture here. So we presume that the bush pigs are the source of this component of the crypto that we're seeing in the system. And then when we do sequencing on the cryptohominis that we find in the chimps, we find that it is actually very similar to an anthropogenic strain. It's a strain that's been seen across Africa in different areas. It's been found in HIV positive children in South Africa. It's been found in baboons in Kenya. So this really suggests that this is a case where this cryptosporidium hominis is coming from people and going into the chimpanzees. We also were able to look at bacterial pathogens in the system. So we looked at things like Salmonella and Shigella, which cause bacterial dysentery. That's a diarrheal disease with a lot of blood loss. It can be fatal. And Shigella is actually really, really important in terms of the health and survival of young children in sub-Saharan Africa. So notably, Shigella is typically thought of as a human-to-human pathogen. It's not, we wouldn't find that in domesticated animals. Salmonella, on the other hand, has a really broad host range. So it's not surprising that we saw Salmonella in pretty much everything, but interesting not in the dogs. And it's interesting that the Shigella was not just in the people, but also in these wild primates. And then ETEC is one of those pathogenic forms of E. coli that we were talking about. And that was found in all species. So in these rural tropical systems, some of these pathogenic strains of E. coli can be a lot more common than they are in developed countries. When we look at the relatedness of the Salmonella isolates, using pulse field gel electrophoresis, we see strong evidence that there's a linkage between the forms that we're seeing in both of the wild primates, the baboons and the chimpanzees, and the local people. Again, suggesting that there's cross-species transmission. And I'm going to go to the next one. And then in terms of Shigella, again, similar types of patterns where it's very suggestive of cross-species transmission between people and these wild primate species. In the system at Gombe, we've also been able to test for antibiotic resistance, both looking for whole genome sequencing based results and trying to look at this relative to specific pathogens, as well as like through the whole fecal sample. So we're seeing kind of all the resistance that's there, as well as pathogen specific resistance, which is an interesting way to look at this, to try to understand where the linkages are. And what this is showing us with the Salmonella is that the human to non-human primate linkage is more important than the domesticated animal linkage. So we're seeing a really different pattern between Uganda and Tanzania in terms of how people are interacting with their domesticated animals. The overlap in disease transmission between people and their domesticated animals is much lower in Tanzania, but we're seeing a lot of evidence of transmission of pathogens to chimpanzees and baboons from people that are living in overlap with them. So we're now working with the government of Tanzania and with the Jane Goodall Institute on their long-term conservation plan for the region to think about different activities around the national park, places that the chimps go, places they don't go, where to have optimal coverage to reduce disease risk and improve things for the local human population as well. Another place where we've done a lot of work on these issues is in Madagascar. We work with the Center Valbaio. I'm a member of the board of the center. It's an amazing research station that's in the rainforest of Madagascar. And over the last 25 years, Center Valbaio has been working around Ranamafan National Park. And all of these little symbols that you see around the park are different activities, different types of outreach activities that they're involved with. And some of these are health. So they have a health team that's going into these roadless areas and providing a mobile health clinic. And we've been able to work with them, to partner with them to also look at health of people and health of wildlife. So this is kind of the scene. You can see the main building of the research center, where we have really good laboratories to be able to do some of this diagnostic work, and a lot of hard hiking. This is just a picture of some of our team members in Madagascar to give you a sense of the kinds of skill sets that we need to answer these questions. You can see a lot of different kinds of degrees, from MDs to DVMs to MPHs to Masters in Development Practice to Masters in Nursing to physician's assistants to anthropologists to geographers to ecologists. To really do this kind of work for conservation or global health, you need broad skill sets and you need cross-training. You need people who have the ability to speak across these fields. And in this system, we see evidence of lemurs, the really unique endemic primates of Madagascar, being impacted by human pathogens. So this is just one example of this. Entamoeba histolytica is a protozoan that causes dysentery. And it's troubling that we're seeing this in multiple species, including critically endangered bamboo lemurs. And we're seeing it specifically in association with proximity to humans. We have similar results that look at bacterial pathogens and also for viral pathogens. And so this is a big concern. But as I mentioned earlier, we don't always know if these pathogens are actually causing disease in the wildlife. Just because they cause disease in us doesn't mean they will cause disease in other species. They may, and that's what we're concerned about. But how do we try to better understand that? How do we figure out if they are actually suffering because of these infections? Well, we're developing a novel assay to investigate that so that we can actually see if they're mounting an immune response. Because as I said earlier, we can't just go and get a blood sample from most of these guys. And that means we have to figure out non-invasive ways to understand if they're mounting an immune reaction. One of the exceptions to being able to get a blood sample are the mouse lemurs, the smallest of the primates. They will readily have a heart trap to get a banana or some peanut butter or a little dried fish. And because of that, we can get a blood sample from them, a very small blood sample, as well as a fecal sample. And that way we can optimize this assay relative to the traditional way of measuring immune function through blood. And we also have the reference genome for this mouse lemur. And because of that, we can actually look at conserved genes for immune function that they have, that we also have, because we know those linkages very well in ourselves. So we're doing that, and then we're measuring their RNA sequence profiles from the mucosal cells that slough off, the intestinal cells that come off with the feces. We can then see if they're actually transcribing in relation to some of these immune factors. And so that's been a really promising approach to actually get beyond, are they infected or not, and really seeing if they're becoming ill because of this. I wanted to take a moment just to describe some of the work we do with other organisms beyond primates. So Johanna Salzer is another former PhD student of mine and a former DVM student of mine. I taught in veterinary medicine prior to moving to Emory. And so she worked with me there at the University of Illinois on her DVM, and then at Emory for her PhD. And she's now in the special pathogens group at CDC. But for her PhD, she examined how anthropogenic disturbance in the form of habitat fragmentation and logging, selective logging, how those anthropogenic factors alter parasitism in mammal communities. So she looked at all the wild mammals living in these forested areas that were being impacted, and how these activities altered the dynamics of their natural pathogens. So we can talk more about that for those of you that are interested. And another example, a project that we're doing right now, is looking at the same kinds of dynamics with land use change, anthropogenic disturbance, and bat disease dynamics. And also looking at this in relation to some of the ectoparasites that they have that could actually serve as vectors for some of these parasites. And this is work that we're doing with my current PhD student, Amanda Vicente Santos, who's from Costa Rica. And we're doing this work in Costa Rica along with the University of Costa Rica. So that's another project that we can talk more about for those of you that are interested. The last thing I want to cover today is, again, what can we do once we have this information to help protect species? I've talked a little bit with you about my work with the IUCN. A big part of that is actually working to protect wild primates from disease. And a few years ago, we developed best practice guidelines for both health monitoring and disease control in the great apes. And these guidelines provide the best practices for us to prevent us from infecting the apes, as well as other people involved in other activities. And then how do we monitor them to figure out if they are being exposed in some way, or if their natural disease dynamics are changing. And then, if need be, how should we intervene? When should we bring veterinarians in to work with specific individuals that are being impacted? Some of the key aspects of this are things like where we're in proximity to these threatened apes, keeping a distance, a minimum distance of 7 meters, being healthy and vaccinated. And also this relates to having an employee health program to ensure that people who are engaging with the wild species are healthy and vaccinated. And it also brings about the importance of thinking about community-based efforts. How do we work to improve public health in human communities that live in overlap with endangered species that they may serve as a source of disease for? With the COVID-19 pandemic, we actually found that the guidelines that we had provided, although they helped in many ways, were not enough. And we needed to develop further information to guide individuals to keep the risk low for us potentially transmitting this virus to wild primates. And so we've developed a number of tools for that and publications. And we've created the Non-Human Primate COVID-19 Information Hub as a real-time source of information on what's known, what's not known, and what's needed. And so this is something Emory's a partner along with Stanford, the University of Minnesota, and a number of other organizations. But primates, as many of you know, are not the only species that could become infected with SARS-CoV-2. In fact, we know captive mink have become infected, and captive tigers have become infected, and groups are concerned about the potential risk of spillover and spillback with bats in areas outside of the range where the virus originally emerged. And so the IUCN has also developed a broader range of guidance for dealing with the SARS-CoV-2 virus in all wild mammals. And so we're trying to ensure we don't expose wild mammals around the world. And so these are just basic exposure risk guidelines that relate to contact, aerosol exposure, and environmental exposure, as well as mitigation strategies related to minimizing, assessing, and protecting. And so this is sort of the state of everything in relation to these dynamics. And this is an area where there's a tremendous amount of work happening right now.